Okay, great. So before we begin the class, I would like to introduce myself first. And my name is Hadar, and I am married with 2 kids, and also have a very nice Shih Tzu dog named Cloud. And I did all my degrees actually in everything in Israel, in Tel Aviv University. I did my degrees, bachelor degrees in physics and biology, then masters in biotechnology, and eventually also a PhD in electrical engineering. So I kind of went through different disciplines because I find it very curious, all these kind of subjects. And then I went to the United States. Over there, I did my postdoctorate fellowship at the University of Maryland. And I'm focusing on, my research focuses on developing miniaturized devices, small micro devices that can continuously and very fast can detect different types of molecules in different types of biological fluids, such as blood, urine, sweat. And this kind of expertise led me to this, to explain or to establish or to propose this class for you. The research in my group, we work on developing functional bio-micro devices. So there are different types, and you will learn more about these kind of devices in this class. For example, how you can develop technology that's called lab on a chip, doing all the processes in a laboratory, commercial laboratory, directly on a small chip. Different types of multi-sensor arrays, and understanding the interface between microelectronic devices and biological molecules. And in that, we study all kind of molecular information. These are what kind of levels or concentrations of molecules directly from the organ level, from the milliscale, down from the cell level and to the molecular level, which are, for example, DNA, metabolites, or drugs. This is my group. We're a very versatile group, and we are part of the Ilsa Katz Institute for Nanoscale Science and Technology in Bengal University. And where is Bengal University? Well, I guess this is China, and Israel is the small dot here in the middle. Yes, we are very small, about 8 million people, almost 9. And this is Israel itself. Be'er Sheva is here. It's kind of in the middle of Israel, although it's a bit here, it's a bit more to the north. This is how very nicely campus. The good thing is Be'er Sheva, it's mostly warm and nice. And those that don't know, where Weber did his postdoc in Haifa, which is also a great place. So everyone are invited when it will be possible to come and visit Israel. Now, Ben-Gurion University is called upon Ben-Gurion. Ben-Gurion, David Ben-Gurion, he actually was the first prime minister of Israel. And he had this kind of crazy idea that, he said that whenever he wanted to think hard, he wanted to be creative, he was standing on his head, and it helped him to think. So obviously, I guess, when I want to be creative, I don't stand on my head, but the good thing is that, Ben-Gurion University, we are very much pushing towards excellence and creativity. Let's lead me to the last slide of the introduction, is that, we have open positions. If you're interested, I guess, look in this kind of class or thing that we are doing in my lab, my group, We have always open master and PhD positions. There are actually a lot of scholarships for Chinese students. And I put this kind of information, and everyone who is interested can ask more or through email or later in the class. So we are in the class for biosensors and lab-on-a-chip microsystems. And this is the first lecture. It's introduction to BioMEMS. Now, BioMEMS, it's a long word. It's an acronym, abbreviation of a long name. But we will learn more about what is this kind of BioMEMS. Now, usually, it's good that I see everyone. So if anyone have any kind of question during the lecture, is mostly welcome to raise their hands. I see you, and I guess also hear you, and you can ask the question. I very much encourage people to ask questions. So imagine for yourself, you know, you're you're a soldier in the terracotta army. Beautiful place, I have to say. I was there 3 years ago. Beautiful place. But you think about it, you are living as a soldier in the terracotta army, but you're feeling not very good, feeling a bit ill. How would you think that during these Middle Ages, during this many years ago, how would you think that a medic or a physician or clinician could diagnose? What would you have? What ill do you have? I mean, those times we clinicians didn't have any stethoscope, any thermometers, any kind of this kind of advanced modern technology that we have right now. For example, yes, one of the soldiers that the clinician is testing, he didn't feel very well, but when the clinician came and looked at the urine of this patient, he saw it was very intrigued to see that there's small ants. Ants were attracted to the urine of the patient. Now think about it. The only way for the clinician to diagnose during these days, he didn't have any thermometer, anything. He just looking at the urine and saw that the ants were attracted to the urine. And that's led him to think about, which later on led to was the first diagnostic patient for diabetes. Diabetic patients their urine has a lot of glucose, and it's very sweet. That's why the ants were attracted to the urine. Does anyone have any questions for this slide? Okay. Whether everything is okay?
Yes, you can continue because the students say they only understand a little. So I will write the key points on the blackboard, so you can continue.
Okay. Later on, it was described by a physician called Thomas Wheeler, that he diagnosed diabetic patient that suffer from basically a challenge, or they are unable, they don't have the enzyme insulin to catalyze glucose. He said that Over there, they were tasting the urine of the patient, really tasting it, and said it was wonderfully sweet, as if it were imbued with honey or sugar. So yes, was then when they were tasting the urine of patients. Thanks God that for today, we don't taste any urine to diagnose diabetes. We use the glucose meters when we take the finger pricking the finger and tasting the blood. But think about it. You know, if today we test this kind of diabetic patients by pricking the finger and testing the blood, what could be happen in 60 years, 100 years, 300 years? Could we think that our diabetic patients or any diseases will be diagnosed with a small micro and nano devices that can continuously flow in our body, detect different diseases, and maybe can automatically administrate the treatment? So this is all about this kind of class, and here we're gonna learn how this kind of functionality of detecting a disease can be done with microelectronic devices, small devices that can continuously or detect this kind of diseases directly in the biological fluid outside of a commercial laboratory. And this is the intermediate phase from the Middle Ages diagnosis into the nano scale diagnosis. So the goal of this class is, and for you also, how you're going to succeed in this class is, the goal is for you not only to learn about, about the theory or what are lab-on-a-chip devices, but also to develop cognitive tools for better learning. And the way for that is, yes, to participate in the class. It's shown, the research have shown that students learn much better and understand the material by asking questions, by talking to other students about the subject, and eventually enable a fruitful discussion between everyone. The syllabus of the class, we have 8 lectures. The first one is this one about introduction to bio-MEMS. And here you see for the first time what is MEMS, microelectromechanical system. We'll learn more about it soon. The second and the third lecture will focus more about the technology, the system level technology of lab-on-a-chip and microfluidics. And then we will go down in the next 5 lectures on what happens in the molecular level, in the physical and chemical reactions at the molecular levels between biology, molecules, and microelectronic devices. We learn about electrochemistry and how we can use electrochemistry to detect different types of molecules. We learn how to design and simulate these lab-on-a-chip devices, the micro devices, and eventually we also learn how we can manufacture these kind of devices. There is a recommended literature, and I will also send it in the widget, that there is a list of all the recommended literature per lecture. Over there, you can go inside and look for the relevant pages that summarizes or explain more about every subject, because this is a very, I cannot explain everything in the lecture. Therefore, I very much recommend to go into those kind of books. They are available, and read more about it. And if you have any questions, you're mostly welcome to write the questions in the chat group. Okay, so until now, does anyone have any questions? Everything is clear. Do it with your head, then I will see. Yes, no? Everyone that understand, do like this.
Yes or no？大点声音，如果他没有问题的话，可以大点声。
Okay. So what do you say, Webby? Should I repeat it or is it clear?
Yes. Clear，你可以说大点声音，如果听。
Okay, maybe we ask questions more in the and continue. Okay.
Actually, they already say clear and so you can.
I didn't do like this.
Yeah.
Let's say that if you understand, do like this, and if not, then like this and ask questions. Okay. So the goal of this lecture is we're going to define what are microelectromechanical systems, those MEMS thing. We see how we can integrate biology with MEMS devices, and then we're going to see some examples of MEMS devices and applications. Now, you see this image here. You see a person doing some physical exercise, and there is a small black thing. You see it here on his arm. What do you think that this kind of black thing is? And here you need to answer me. What do you think this kind of thing is? Don't be shy. There's no mistake.
Sensor.
Sensor. Very good. It's a sensor. What does it measure?
Force.
Force. Okay, force. What else? This is interactive course. What is it? Neural activity. Very nice, very nice. Okay, so we said force, neural activity. Remember this kind of thing. At the end of this lecture, we will see what it is. So what are MEMS? MEMS are the acronyms of microelectromechanical systems. It integrates microtechnology, nanotechnology, microfabrication, how you make this kind of, how you can make small devices the micro scale, and microsystems, how you can integrate multiple systems together. Examples for MEMS devices are, for example, electrostatic micromotors. You see kind of small, there are those kind of circle devices. We have micromirrors, tunable inductors, all kind of these kind of devices were manufactured at the microscale. Now, microfabrication or microfabrication technology is a way to make stuff. We fabricate, we make, manufacture. And it was adapted from the semiconductor industry. You know, Intel or different types of semiconductor devices. It allowed different types of materials and way to shape them. And this kind of the technology that was done for the last 50 years was enabled to make small devices that are functional, that can do different types of activities. For example, these kind of very small devices, but they have some challenges. For example, because they're very small, suddenly a mite or this kind of biological organism can be on the size of the device and it can damage the device. But also, for example, this is coagulated red blood cells, multiple blood cells, red blood cells that are connected together, and they can actually damage the activity of the micro device. All of these are 50 micron size. So what are MEMS? Again, we are going to look at MEMS from different angles. The definitions varies. Usually it's made, or MEMS devices are, again, devices that can have some kind of activity, functionality. They are made from the semiconductor fabrication industry. We use technology that is used, for example, to make microprocessors, but we use the technology to make these kind of different devices. So we harness technology that was used to do semiconductor devices such as processors use it to make those devices. Usually small, smaller than 1 millimeter, and has some kind of functionality, activity it's used for. Now, there are two types of MEMS devices. There are sensors. It means there is some kind of information in the environment or in biology, for example, such as you said, neuronal activity. And we want to use the MEMS device to translate this kind of information into electronic data that we can measure. So this is the direction of information. Now, there are another type of MEMS devices, which is the other way around. It's called actuators. And now we use electronic data signal and translate it into actual biological or any environmental activity. For example, we can use different heart stimulation. Heart simulation, use a device, you apply energy, and it affects the environment, the biology, the heart itself. This is type of an actuator. So sensors, direction from the environment into electronic signal. An actuator is from the electronic signal into the environment. Does anyone have any questions on this slide?
没有 question 就这样比个手势，如果有就是朝下。
Ah, great, great. The guy with the black hat already understand. Very good. Okay. What are MEMS? Now, we said microelectromechanical system. So it's system, micro devices that have both electronic activity and mechanical activity. But it doesn't have to have only also both electronic and mechanic. It could have devices that don't have electronic activity, for example. So such as static microfluidic structures. You can flow a solution through a micro scale channel without any electronic manipulation, just by physical activity from higher pressure to lower pressure, but it's still a MEMS device. Or you can have a MEMS device which doesn't have the mechanical part. And here it's a valve that based on two electrodes that allow heat one area of the channel, and therefore because of due to different gradient of temperature, the solution flows from the different temperatures. So we can see that they have different types of MEMS, but we need to understand the physics in order to invent two new types of MEMS devices that do different functionalities. Why small? Well, we want to do micro devices. We can do small devices. First, to save time and cost. We have small devices, it's less material, short processing time, so we save time and money. It's usually disposable. When we do small devices, the material is very cheap, and we can throw this kind of device later on. We don't need to do multiple times. We can also do multiple processing for the sample, and we can integrate, because we have small devices, we can do like a puzzle. We can integrate, we connect different parts that have different functionalities to do automations. And there are very nice physics that happens in the microscale that we're going to learn in this kind of course. For example, laminar flow. It's a phenomena that when you have a micro flow in a microscale, the flow, you don't have any turbulence of the flow. Or we have things called high surface to volume ratio. We have a very high surface area, but with a very small volume, meaning we can have very rapid heating, for example, or the high signal to noise ratio, and so forth. We're going to study this kind of different types of physics, but eventually this is open area. So whenever you understand the physics, you can also think about new types of devices. So small MEMS devices, usually from 1 millimeter into the 100 nanometer size, from a hair, which is around a diameter of 100 micron, red blood cells, usually about 10 micron size, and HIV, or even the, now it's very trendy to say the COVID, the SARS virus, into the 100 nanometers. So we can also think about, and I'm going to discuss it later, how we can make those MEMS devices to detect, for example, the COVID or the SARS virus. Now, what kind of devices we know? MEMS devices. The very known one is the, if you are familiar with the Wii or Nintendo or any kind of device or video game, that you have those kind of sensors in your hands and you can move it. So it's based on the, and so this is the MEMS device that allows the recognition of the movement. It's called Accelerometer to measure the acceleration. So it measures the acceleration in the X, Y, and Z direction. And from the movement, it translates the movement from mechanical side into electrical signal. And the electrical signal is being analyzed by the system, by the further electronic analysis, signal analysis. Or the inkjet printer, how you can make a very small drops with a very small dimensions. So that's also used with MEMS devices that uses resistors in order to enable a bubble size of a drop to print, the ink. Now, this was MEMS devices. MEMS devices, we haven't talked about biology yet. Now, bio-MEMS devices is taking biology and integrating that on a MEMS device in order to have a more interesting functionalities, biological functionalities. And this is the main thing we're going to focus on this class, how biology can be integrated on MEMS devices to enable new functionalities. So bio-microelectromechanical systems, we have different types of technologies, such as the microfluidics, and this is going to be the third lecture. Biosensors and bioelectronics, how we can detect different types of biology or biological molecules. Neuronal interface devices, how we can interface neurons. We can also use bioreactors. Doesn't have to be sensor detectors. It can be also bioreactors to generate or to use biology or cells to generate, for example, medications. Bacteria can be genetically engineered to produce medications in a very fast way. And also implantable devices, drug delivery devices, small devices can generate medications in implantable devices inside our body. So one of the technologies that are, as I said, is microfluidic. Microfluidics allows us, it's basically how we can flow liquid in microchannels, and we can use this kind of technology for different types of applications. For example, for high throughput analysis using droplet technology. So what it does basically, we have different like two channels from here and two channels that allow solution to go inside, and one outlet channel. So what basically happens, and here you can see with the color, is that you have two different colors goes here, and from the other side of the channel you have oil. Here you have water type or aqueous solution. Here you have oil. So with the kind of velocity for the side channels and the middle channel, the solution is being isolated with two oil areas. So basically, this kind of technology allows us to manufacture droplets or nanoscale or picoliter, nanoliter or picoliter, 10 to the minus 12 liter volume droplets. You can see it nicely here. And you can do anything, whatever you want to do in kind of droplets. You can do different types of biochemical synthesis. Okay, you can introduce, for example, enzymes and substrates and put them together in these kind of droplets, and these droplets would make some kind of biochemical reaction. Or you can do, for example, a PCR, digital PCR. What does it mean? For example, you can introduce the sample for the COVID virus, put the sample here, flow it, and from here, add all the cofactors, and here will be the reaction of the PCR multiplying the RNA, the virus code that will detect if, for example, there was some virus code or there was some SARS or COVID in the sample. The reaction that will happen here will be such as fluorescentic, and we can detect some light from that. So again, we can use it, technology, for different types of applications. And here, I like this kind of video. You can see how we can use droplet technology and to flow. And you have inside those droplets, you have a cancer cell. And the idea is that if you have the reaction with the cancer cell, you will have this kind of black colored droplet. and it will be more charged. So whenever you have these two electrodes, they will generate specific repulsion forces and will force the cells or the droplets with the cancer cells, which are more negatively charged, for example, to go into this channel, and those that are not charged, you don't have cancer cells, they will go to this kind of area. So here, this kind of, again, we can send the physics and we integrate electrostatics together with flow and biochemical reactions. And it's very cool. You can do, I think, about 6 million droplets in one hour. See, their sorting rate of 2,000 droplets in 1 second. It's crazy. This video was very slow from the real time. Crazy. Now, more examples. You can use a thing called the Electrophoresis cell sorter, which means that if you apply electrical field with an AC signal, it causes cells to have a specific polarity. Polarity has one side which is positively charged, the other side of the cell has negative charge. And then you can apply some kind of potential and cause the cells to flow, depends on the polarity. So I'll give you some examples. Analysis of single cells. You can use or build devices that can analyze one cell. So we talk about neuronal activity. So this group of Ambrecht and Dietrich, they fabricated those nano electrodes that now when the cell or neuronal cell can sit on these kind of electrodes, and you can detect or you can measure the electrical activity from the neuron cell, from a single neuron cell. And you can have an array of them. So basically, you can measure the neuronal activity from different neurons simultaneously. It's crazy. More example, DNA microarrays. You can make or manufacture, and this is an example of the company it's called DNA chip of Affymetrix, company called Affymetrix. And what they did is that each dot that you have here, you have thousands of dots. Each dot has a single type of a DNA strand. Now, a DNA strand, this is our code, our genome, a DNA strand with a single genetic sequence of a gene. So each dot has a sequence of a DNA with a different gene. So basically what happens, we can take a sample from our body, extract the DNA that we have, and put it on top of the chip. That way that wherever we have a gene that is the same or complement for the one that we have in this kind of different spots. And then we do another process that whenever we have this kind of DNA hybridization reaction on a specific spot, it will generate fluorescent response. So whenever you have the highest response, it means that we have genes in our samples. The genes are the same. And when we don't have any fluorescence, so the sequence that we have in this kind of area, we don't have these genes in our sample. And you can do it for thousands of genes simultaneously. It's a small chip, and this is how it looks like. There's a lot of work of image processing to identify or to make this kind of images and to analyze them to understand exactly what happens in each one of the spots. Another example is PCR, polymerase chain reaction. For those who don't know about, I guess you're familiar or you heard about the definition PCR, usually being used for the COVID tests. Before you leave, people ask you, okay, you need to show the proof of a PCR test that you're negative to COVID-19. And that's usually done by, you have the virus, and the virus itself has proteins, kind of like a capsule of proteins, but inside you have the RNA. The RNA, it's a sequence of the genome of the virus itself. Now, usually you have, one RNA sequence, one RNA strand for a virus. But you want to detect a very small amount of viruses. So you have a very small amount of RNA that you cannot detect. Current devices cannot detect single-stranded RNAs. So what they do, and that's why it takes some time, you take the sample, the swab from your mouth, from your nose, put it in some kind of solution that removes all the proteins that cover the RNA. And now this kind of RNA is done through processes that takes the RNA, it copies the strand and divide it. And then those two strands now being copied again into 4 strands, and so forth, from 4 to 8, and from 8 to 16. And that's multiple cycles, take a couple of hours. And until you have enough copies of the RNA of the virus to generate enough signal that now the test shows, okay, you have positive, or it doesn't detect anything, and then it says, okay, you don't have any virus. This kind of process called polymerase chain reaction, or cycle, continuous or multiple cycles, but it takes time. It takes at least 6 hours to do that. And also you need a commercial laboratory to do that. So for example, if you want to have or develop a device that can do that directly, in the airport or in the entrance to the university, for example, and can do it very fastly, there are devices called micro PCR. And over there, they do all the process of taking a sample, replicating the RNA strand, separating, replicating again, separating, replicating again, all of that done automatically in a micro scale or a small micro device continuously. And because we have very small sample volume, that can be done very rapidly. So usually this is can be done in about 15 to 30 minutes to get enough copies of the RNA in order to know if you have enough virus, for example. So this is the next technology that's currently being applied, trying to replace the conventional PCR. Does anyone have any questions so far? Everything clear? Okay. Other examples, neural probes for implants. So if you can make those micro devices and design them to interface with neurons, we can maybe use it as a thing called brain-computer interface. Brain-computer interface, it's devices that translate the activity of the neurons into electrical signals. So for example, there are people that cannot move that don't have any, has prosthetic hands, and they want to operate them. So they can have, implants in their mind, in their brains, in specific areas that control the motor activity of the hands. and they can record the electrical stimulation from this area and translate with the devices into a prosthetic hands and move them according to the electronic signals. There are many challenges there. It's a fascinating area. Very, very cool. Tissue engineering. You can build devices and control the growth of the cells in a very, very controlled way to enable a tissue that have different functionalities. For example, this group, Poleo, back 2009, they made a device. They grow cells of the epithelium, apical epithelium, which are the cells of our eye, of retina. And they grow it on a device and use it. Now they can take this kind of layer of cells and use it as an artificial cornea and implant that on the eye. And that actually was done. It was done first on rats, but now there are actually company also in Israel that make those kind of artificial corneas that can replace damaged corneal from patients. But don't always have to grow the cells. You can also have these other types of sensors. This company called Sensimed, what they did, they developed a sensor on a land, an island, that can detect glaucoma. Glaucoma is a disease that has some under pressure, ocular pressure in the eye and damages the sight. So they built this kind of sensor that can put it on the eye like a contact lens. And the sensor, it's actually this kind of lines. These are gold lines that measure the capacitance between the line. So you have two lines, like a capacitor. You measure the capacitance between them. So whenever there's some change in the pressure, so the curvature, how much curvature of the eye it is, changes, and the distance between the electrode changes, which affects the capacitance. And this capacitance is being translated into electrical signal, and in this kind of black part, the information is submitted wirelessly to the device itself, to the phone, for example, to detect glaucoma. Now, in the last 15 minutes that I have, I will talk a bit about how you can use this kind of micro devices, bio-MEMS devices, for point-of-care testing. So there's more and more, whenever we don't feel very well, we go to the physician, they do like a blood test, send it to commercial lab and tells you, okay, this is how much white blood cells or specific protein that you have, and this is that's why you're sick. However, this kind of process of taking the sample, freezing it, transferring it to the commercial laboratory, all of these kind of steps actually changes the composition of the molecules that you have in a sample. So whenever you measure that in the commercial laboratory, it's actually have different molecules, different concentrations than the one when you took from the body. Therefore, the analysis or diagnosis will be not very accurate. So recently it's been done an area called point-of-care testing, meaning that we can bring the test from the commercial laboratory to where the person is being treated. And think about it like if you are in the physician office, you can take a test, do that in his office and automatically get the results, or maybe at home. And with that, you can get a more accurate results and have a more called personalized therapy, a treatment that is more personalized to your condition and not to the general society. And you can use paper as a substrate for the devices. You can use array of sensors, small microbeads that can use for sensor and microfluidics. We'll talk about it. Can you take protein, RNA, DNA, cells, metabolites? And we need you, young students, we're very creative and bright, to think about new types or new strategies to sample the clinical sample, to analyze it, to analyze the data that's being generated, which eventually will have new ways to do point-of-care testing in on-site disease diagnostics. Now, in this class, we're going to focus on a type on sensors, on bio-MEMS, which are sensors or biosensors. We'll learn more about biosensors also in the next tomorrow. But for that, first, to know what a biosensor is made of. And it's composed of three parts. A biosensor made of a physicochemical transducer, a receptor, and the output signal. These are the three parts of a biosensor, and with these parts, it can detect the presence of a molecule, or we call it a marker or a biomarker. So the receptor part, such as antibody, enzyme, it can, or DNA, it binds to the molecule itself. You see, it's kind of like fits well. So the receptor allows a selective detection of the molecule. It fits the molecule itself and not to other molecules you have in the sample. It's very selective or specific to the molecule itself. That's the goal of the receptor. Now you have the physicochemical transducer, that its goal is that whenever there is a binding between the molecule and the receptor, it can translate or transduce this kind of binding into an output signal. And the signal, output signal, is the third part, and this is the signal that is generated from the binding, and it could be optical, electrochemical signal, mechanical signal, or different types of signals. And the idea here, and we will return, we will get back to this kind of scheme many times during the class, during the classes, and we're gonna analyze different types of biosensors or bio-MEMS devices. And we're going to say, what is the receptor, what is the physicochemical transducer, and what is the output signal. And this is very important because in order to get more experience in bio-MEMS devices, it's not about studying the book, it's about analyzing as much as you can biosensors in order to learn more about what the options are. Do you have any questions about this slide? Okay. Now, in order to design a good bio-MEMS or biosensor, we need to know very good about what we want to detect. And here comes the need also to learn or to know about the biochemistry, the biology, not only about the physics or the engineering parts, but also to understand how the disease happens. Or in the case of the COVID, to understand how the virus looks like, what are the proteins, what are the RNA, what is the sequence, to learn about how the biology behaves, and from that to choose, okay, I want to, in order to know if the virus is present, I want to detect the RNA, or I want to detect the protein that, that covers the RNA, and so forth. So in order to know or design a good sensor, you need to know the biology also. So some examples for biosensors for point of care testing. We have a sensor that is made of paper, regular paper, different levels of papers connected together, but each level has a different kind of a treatment or different analysis. So the idea is that you take, you prick your finger, and again, the goal here is to detect liver function. So liver function is the behavior of different types of metabolites. The liver metabolites compounds. So you take a pick of a finger sample, you apply it on the device. Here's the blood, and in the endpoint you have the reactions of the sensor, and the different colors or the intensity of the color tell you, okay, how much enzyme you have from ALT. It's a type of an enzyme. Okay, or this color tells you, okay, how much you have from that. And built from different layers of the paper, and let's see how it builds. So it has the upper level with a small hole inside. This is the upper level. So basically allows the blood to go from this area, then goes to this area. And here you have hydrophilic or hydrophobic paper that allows 5 different chambers that has different types of enzymes. And then it's connected together with the bottom part that doesn't allow to water to the sample to go down. And when you connect it together, you put the sample, it goes react with the enzymes, and you can read the reaction from the bottom. So here specifically, if we think about back again about the components of the sensor, we have the receptor, the physical-chemical transducer, and the signal. We always start to analyze from the output signal. So from the output signal, we see colors. Colors means that we have optical signal, output signal. Okay? If it was electronic signal, we can say it's an electrical or electrochemical signal. But here the output is optical. We look with the eye and see different colors. So the output signal is the optical signal. The physical-chemical transducer, okay, the one that it translates that into color is the reactors, the molecules that we have there that generate the color. This is the transducer, and the receptor that detect the enzyme are the basically what we have inside that are bound inside the chambers, and they are the if you know, an enzyme has substrates that it generates products. So the substrates for the enzyme are the receptor. So it means that the enzyme is there, okay, but if we don't have the substrate, so we won't get any reaction, we don't get any color. So the substrate is the receptor for the enzyme itself. Other examples for point-of-care testing devices, we can have disposable devices for such as pregnancy tests, or wearable devices we can wear on our on our skin. We have smart lens with our sensor that we said from the that actually can detect glucose in our tear for diabetic patients. We can electronic skin, and this is actually something that maybe I can need to update that into Professor Hussam Haik work also from the Technion. We can have handheld blood analyzers. So we can have a small drop of blood, put it on a cartridge, and detect different types of like sodium, potassium, chloride, and so forth, different types of metabolites we have in our blood. It's basically very simple. You buy this cartridge, you put the blood, put it inside the reader, and after about 15 minutes, you can see the results. Point of care testing devices, it's a huge market. Only in the US in 2017, we had 4 main companies, Roche Diagnostics, Abbott, Bayer, and Johnson & Johnson. And only for the glucometers, sensors that measure glucose, we can see that these numbers are in millions of dollars. So only, no, it doesn't make sense. Yeah, millions of dollars sales for only this device for a year. So it's billions of dollars market for different devices, and it's only for the glucometers. You can think about other devices can detect other types of molecules. The most known one, we talk about the glucose sensors. So there is the continuous glucose monitoring CGM devices from Abbott company called the Freestyle Libre. It actually is this kind of device. It's underneath the skin and continuously measure glucose level in the interstitial fluid. It's the fluid that happens below or between the blood vessel and the skin. And you can read it with your device. Very important device. It really revolutionized the way diabetic is being monitored. You can also implant those devices in the body. And this company, Sensionics, developed a sensor that can implant it in the body and can continuously measure glucose levels in the body. And you don't need to put the device beneath the skin, just implant it. And with that, we can see that there are many types of examples, and the area just going in smaller and smaller. Did you have any questions so far on different devices or anything that wasn't clear? There are any questions? I don't see. Okay. So with that, I'm concluding the first lecture, and the goal was to understand what are MEMS devices, and most importantly, see what are bio-MEMS devices, understand what are biosensors, and show different types of applications that can be used for bio-MEMS technology. And it was only the tip of the iceberg. We can see there are many papers going, many examples. Every day you have new examples. So in this class, I'm gonna try and expose you to some of the examples of the recent examples. And eventually it's up to you to look for new papers, for new examples, and to expand your expertise. So now we just summarize this lecture. And if you want to identify a biosensor, and now we're gonna see 3 different types of devices. We're gonna see the device that measures cholesterol in the blood, a device that measures the EKG, our heart functionality, and a device that measures CO2 gas. And I want everyone to look at me. Look at the slide. Raise your heads, and I'm gonna ask you, who thinks that the device that measures cholesterol is a biosensor, according to what we discussed. Remember, a biosensor has an output signal, a physical chemical transducer, and a receptor. So who thinks that number one is a biosensor? Raise their hands if you think so. Don't be shy. Don't be. Who doesn't think it's a biosensor? Okay, I feel only one that don't think it's a biosensor. So number one is a biosensor. It measures molecule, a cholesterol in the blood, and the output signal, you can see the electronic screen tells you how much you have cholesterol in the blood. So this is a biosensor, has a receptor, physicochemical transducer, and an output signal. Now, number two, who thinks it's a biosensor? Raise their hands. Okay, we see more hands. Who doesn't think it's a biosensor? Raise their hands. Okay, so this is kind of tricky. This one is not a biosensor. According to what we discussed, it doesn't measure a molecule. It measures electrical activity. So remember, when we talk about a biosensor, we want to measure molecules. And here, this EKG, electrocardiographic device, measure electrical pulses and not molecules. So it's not a biosensor. Now, number three, measures CO2 gas. Is that a biosensor? Who thinks it's a biosensor? Raise their hands. Okay, the guy with the red shirt. Who doesn't think it's a biosensor? Okay, so the right answer is that we don't know. What does it mean? It does measure a molecule, okay, but we don't really know how the device is working, because basically, the physical chemical transducer with CO2 could be different types of transducers. They could be sorry. No, sorry, I'm taking a step back. Sorry. This is a biosensor, like we discussed. Sorry, this is a biosensor. It measures molecule CO2 and translate it again into electronic signal that we can know if we have the CO2 or how much, or if we don't have the CO2. So again, biosensors are 1 and 3, and they measure molecules and translate the presence of the molecule into electronic signal, into a signal that we can measure and interpolate. Now remember that some students said it's measure force or measure neural activity. This work was done, it actually measures lactic acid. It's a molecule that is being secreted in our sweat when we do physical exercise. So here it's a biosensor that measures lactate. And you can see a lot of different examples also from the Professor Hussam Haig group in the Technion that measures different types of molecules using patch sensors on our skin. So this device measures lactate, and nicely it was actually doing that whenever the guy was doing a lot of physical exercise, you see the lactate levels increasing and increasing. It was very cool. Okay, so with that, we are finishing the first class, the first lecture. And tomorrow we're going to learn how we can shrink all the functionalities or the application that we saw in a commercial lab directly on a small microchip, how it's built from different components, how this kind of system is built, and we'll continue from there.